Hello, welcome viewers. So today we are starting a new lecture series on principles of genetic engineering. So in this subject, we are generally dealing with a laboratory based technology which is used for altering the genetic makeup of an organism. So what is that laboratory based technology? So it is called the recombinant DNA technology, or it is known as rDNA technology, which is used for genetic manipulation of an organism. So this rDNA technology involves joining of two different DNA fragments. So to join these two different DNA fragments, you are using one DNA fragment that is called vector. So this vector is the one which generally carry a foreign DNA into a new host system for genetic manipulation. So there are several types of vectors used. So generally a plasmid or phage based vector is used for carrying a particular DNA segment into a host cell. Vector, what are the basic structural units considered? So first one is the origin of replication. Origin of replication is the one which determines the number of copies of the DNA in a cell, or is the sequence which is helping the vector to replicate in a host system. So this is the origin of replication. Second one is the marker gene for selection. Suppose you have introduced a foreign DNA into a host system, you have to confirm that that particular vector is entered into the host cell. So that is possible only by a selection. So usually a marker gene used for selecting such a strain, that is called such a strain. So for that marker gene generally used is antibiotic resistant genes. So antibiotic resistance gene is used. So generally the method what is used for introducing a vector into a host strain, that is called a transformation. So third one is the region to carry a cargo DNA, or you can also call multiple cloning sites. So these three minimal region should be present in a vector: origin of replication, marker gene for selection, and a region to carry a foreign DNA, or it's called a multiple cloning site. So what are the basic factors to be considered when you develop a vector, or when you want to use a particular DNA fragment as a vector? So first thing what you have to consider is copy number. How many number of particular vector is maintained per cell? So this actually determines the performance of the vector. Uh, Suppose if it is a low copy number vector, the amount of protein which is produced from the foreign DNA will be less. So that will not be sufficient to study the genetic effects in the particular host where you have introduced. So because of that, the copy number will determine the performance of the cloned DNA and also the vector inside the cell. The stability. So this copy number actually determines the stability. If the copy number is more, when the cells are multiplying and segregating, in all the cells the copy number will be maintained. And also another thing is, when you consider a vector, all these vectors, for example, when you take a plasmid, at the end of the replication, it is actually segregated into daughter cell by a recombination based method. So that particular sequences for segregation and partition should be present in the vector, so that all the daughter cells will have the vectors passed. Otherwise there will be some host cell will be there where the vector is not present. Daughter cells will be there where the vector is not present. So after a series of generations, you will see the vector is lost from the host system. So because of that, for a stable maintenance of the vector in a host system, you have to put all the structural features which is required for the segregation of the vector inside the host system. The third one is compatibility. Compatibility, you have to see when you see two different vectors are introduced in a single host. Why you need two different vectors? Suppose you have cloned the gene, and that cloned gene you want to express only at a particular time points, or you want a regulation all the time points. It should not be present. Whenever you want that time only that gene to be expressed. So in that case, the control elements you can put it in another vector, and the genes can be put it in another vector. So when these two different vectors are maintained in the host, it should be stably replicating. So after a series of cell multiplication, all the cells should have both these vectors, so that the protein will be stably expressed, or it can be expressed in a regulated manner like this. So the modularity of the circuit, or modularity of the cloned gene will be better when you have two different types of vector in a host system. So the replication of one vector should not affect the replication of the other vectors. So the modularity used for replication of one should be different from the second, so that it can be maintained in a stable manner. So this compatibility also contribute to stability of the vector maintenance in a host system. Next is specificity. When you are using a vector, suppose if you want to clone that or join that foreign DNA and multiplicate in E. coli, and the gene study you want to do it in fungi, so you need to maintain that vector in E. coli and also in fungi. So the vector what you are using should have a broad host range. So when you have a broad host range, the specificity is very important. What are the control elements needed for maintenance in E. coli? That should be present there. And what are the control elements need to be maintained for replication in fungi? That also should be present. So this is the specificity, or the whether the vector is able to replicate only in E. coli, or whether the vector is able to replicate only in fungi. So based on that, you can have vectors like broad host range or narrow host range. So this specificity is also contribute to compatibility. So the replication machinery, when you put it in both the replication machinery, when you put it in a single vector, this should not be mutually inhibitory. So it should be able to, it should be compatible, and it should be able to stably maintain in the host. So this specificity is also depends on the copy number. So this broad host range vector and narrow host range vector, when you use, that also should have a what is the stable maintenance copy to be maintained in E. coli, that origin of replication should be there. What is the stable maintenance of copy number to be maintained in fungi? That type of origin of replication should be present in the vector. So that is the, when you consider the specificity, you have to also see the copy number. So next is the minimalism. So if you can reduce the size of the vector minimum, larger will be the foreign DNA you can join with that. So because, and when you do the minimalism of the vector, you have to consider that the circular plasmids, when it is replicating at the end of the replication, there is a recombination mediated process is there for to segregate the plasmid into two different host strain. So that kind of partition genes, partition elements should be present in the vector when you are using. So you cannot have a very minimal sequences. So what are the minimal sequence to be present in the vector for stability also should be considered. So if all these are considered, you can have a vector for cloning. So when you plan a vector for cloning, you are actually addressing the purpose. So the vector, when you are using, that vector should have a cargo point where you can have different options available, how you can put the foreign DNA in different ways. So that kind of way you can join the foreign DNA elements into the vector in using different methods, whether using either ligase enzyme or without ligase enzyme, or with host ligation methods. So all these things you can consider at the cloning step. So how the foreign DNA to be added into the vector and how the cargo carrying site should be. So those kind of thing you can consider when you're considering the vector for cloning. So, there are different types of cloning vectors available and also which is used in the recombinant DNA technology. So based on biology of the DNA and based on copy number of the DNA and also based on how the insert is placed in the vector. So based on biology, you can find plasmid, phage and artificial chromosomes. And based on how many copy number is maintained, you have replicating vectors, non-replicating vectors. Replicating vectors, when you see, you have stringent and non-stringent. Non-replicating, when you say vectors having transposons, vectors having phage attachment site. And based on how the insert is placed in the vector, based on that, you have insertional vectors and replacement vectors. So in the coming videos, we will be seeing all these category one by one, and how a plasmid is used as a vector, how the evolution of evolution of plasmid vector happened, phage vector happened. So like that, how they are utilized, how a plasmid is used as a replicating vector, or how it is used as a non-replicating vector, or how a plasmid is used as a insertional vector or replacement vector. Similarly, phage and artificial chromosomes like that. So please listen to our next lecture series also for a complete idea of all these principles. I ask you subscribe my channel, share my channel, like my channel, and press the bell icon. Thank you very much for listening to my lecture series. Thank you.